Gene transfer to plants. And first we will start with introduction of plant transformation. Plants, they are very valuable source both for humans as well as for animals. The reason is that they provide food, fiber, and construction material. In addition to that, variety of molecules or metabolites, they are also produced by plants, like dyes, variety of scents, and most importantly, herbal medicines. So because of their value, since centuries, humans have tried to improve the quality of the plants so that they can get valuable products. तो इस पर्पस के लिए इंसानों ने प्लांट्स को मॉडिफाई करने की कोशिश की, like with the help of breeding, and also naturally they have selected the best variety. लेकिन ये जो सारे जो तरीके हैं ये थोड़े से अब ओल्ड हो गए हैं, क्योंकि जेनेटिक ब्रीडिंग से या नेचुरल सिलेक्शन से जो जीन पूल्स है वो पर्टिकुलर स्पीशी तक रिस्ट्रिक्टेड रहता है, या सिर्फ वो स्पीशीज जो कि क्लोजली रिलेटेड हैं दे कैन रिप्रोड्यूस सेक्सुअली. सो बिकॉज ऑफ दिस रीजन now some other methods are required to improve the quality of the plants. And for this purpose, the genes have been introduced to the plants artificially. For this purpose, the first attempt with reference to the transfer of genes to plants was made during 1960s and then later on in 1970s. But all those attempts, they were not successful. The reason was that the transformation was not stable. Then in 1981, the first stable transformation was carried out by the scientist, and here the tobacco plant was used for transformation purpose. और इसकी जो ट्रांसफॉर्मेशन थी वो भी एक प्लांट पैथोजन के जरिए की गई थी, और प्लांट पैथोजन है एग्रोबैक्टीरियम ट्यूमिफेशियंस. इट इज़ अ टाइप ऑफ बैक्टीरियम that can infect the plant tissues when they are wounded, and then by introducing its own genetic material, then it can induce tumorous growth. And this ability of the bacterium has been exploited to transfer foreign DNA or gene of interest. And here the tobacco plant was the first one that was genetically modified by using this agrobacterium tumefaciens. After this success, then in the later decades up till now, more than 100 plants have been genetically modified by using variety of transformation procedures. So in addition to a bacterial gene delivery system, other methods have also been used that we will discuss after some time. But overall, we can say that the ability to transform or modify plants by introducing gene of interest have enabled us to produce transgenic plants. And those transgenic plants, they have opened a new avenue for agricultural biotechnology, so that we now have plants that are resistant to a variety of diseases, pests, and environmental stresses. In addition to that, our gene of interest, it can produce a variety of chemotherapeutic important proteins as well as some industrially important enzymes. When we compare animal with plants, so we can say that plants, they are more, you can say that having more developmental plasticity. Their material, they can be easily transformed by using variety of methods like leaf segments, stem segments, seeds, or undifferentiated mass of cells like callus can be produced from plants that can be used for further modification by genetic means. For all these manipulations, a technique is required which is called as plant tissue culture technique. But no, people have also tried to modify the whole plant, a technique which is called as in-plant modification that has minimized the use of plant tissue culture.